get scammed and tricked into thinking this is real, when it's just random chance that it comes out. Okay? So, yeah, you can still see that. Okay, but I'm just trying to illustrate what's going on with multiple testing here. Okay. Okay, some pedigrees are more informative than others. Okay, this is a less informative pedigree because, say the grandparents die, and you can no longer collect DNA samples from them, or they're not around, you can't do that. You can still infer, using this data, something's going on. So it looks like the 5 is associated and recombined, but you don't have a lot of certainty about that. You gain a tremendous amount of power if you can genotype 3 generations. Because you know the phase, you gain a lot of power and certainty about what's linked and what's unlinked. So this is less informative and less useful, and this is uninformative. You can't use this to do anything. So even if you have the grandparents, if this guy is homozygous, who are the recombinants and who aren't? For this microsatellite, it's useless. You throw it away. You have to move on to the next one and establish linkage. This doesn't contain any information that's useful for you. Okay, so we're not going to do this. We talked about very simple Lod score calculations. If you have dominance at an autosomal locus, you can see everything that's going on, complete penetrance, et cetera, et cetera. In actual applications, you can modify it. You can take into account incomplete penetrance, if it's recessive, and so on, and use lots of markers simultaneously along the chromosome. Okay, so that it makes it impossible to do by hand, but there's software that can do this if you plug all this information into it. So I just wanted to kind of give you full disclosure, but I don't want you to worry about the details of how you actually do that in this class. Okay, so here's an example. A real-world actual example where this has been done. Keratoconus is this disorder with the eye. So the cornea has a different shape and it impairs vision. So you can see this bulge effect right here in the middle compared to a normal eye if we look at the cross-section of an eye. And this is something that's inherited through families. So here's pedigrees of 20 Finnish families used in a linkage study that were affected with this trait. So it pops up from time to time. It looks kind of recessive, but it's a little odd. And this is like real data. It kind of pops in and out and doesn't fit a simple pattern very well. And sometimes you have grandparents and sometimes you don't. And let's see, what else? You know most of this. This refers to twins. These are identical twins when you have that kind of connection. But other than that, I think you've seen all of this. Okay, so you genotype lots of microsatellites in these pedigrees and combine them. And this is what you get. These are Lod scores on this axis and location across the whole genome. So these are the different chromosomes, 1, 2, 3, and so on. Okay, and you have so many microsatellites, you have a lot of information, all these squiggly lines, all along the chromosome. And here, -2 is exclusion. So there's lots of points, like this big chunk of chromosome 3. You're sure it's not there. Okay, you've excluded it. The disease gene is not in that location. It's not in this area on chromosome 9, and so on. It's not significantly linked anywhere until you get here, chromosome 16. There's a spike. Okay, there's strong linkage at that region with this disease state in these families in Finland. So that's the interesting point on chromosome 16. So you can zoom in. Here, Lod scores jump all the way up close to 4, so that's above our very stringent 3 cutoff for linkage. This is the interesting region. These are the designations of the microsatellites. And they went in and typed additional microsatellites just to make sure this was a real pattern and to nail it. And then you can take a look at the known genes that are in this region. Okay, so this gives you a list of candidate genes after chromosome 16. You can take a closer look. Are they involved in development? Are they expressed in the eye? Are there mutants in the gene sequences in these people, in these pedigrees? Okay, so it's not the whole story, but it gives you a way to narrow down and see what's going on and to actually nail what gene it is and what mutations are taking place and how it's involved. So this is what has been done for a while with human genetic studies in mapping these things. We can't do test crosses. We can't use a lot of tools that we use in other species, but you can use pedigrees to do something similar. And in the end, you get to the same kind of result. You map a region that you can test. Okay. Recap. Log scores. So they're just measures of linkage. It's a different way to establish linkage between markers and a disease-causing allele at a gene that you're interested in. Okay? And it's just the base 10 log of probability of the data, assuming a recombination rate divided by random chance, no linkage. And you can combine results from multiple studies. We talked about the caveats. You have to assume to do that. And thresholds of 3 and -2 are the keys to establish or rule out linkage with the disease gene. This is just sort of an afterthought. Some pedigrees are informative and some are not. So you can't always use a pedigree, even if you have lots of data from it. Okay. Okay, let me just pause for a second with LOD scores. I think you know how to calculate them and interpret them based on what we talked about. Any questions before I move into something else? We're good? Okay. Okay. Let's see. Let's back up to a simple question here. Let's see if I can start this. What is the uncorrected genetic distance? So I'm talking about recombinant fraction between white and short, these outcomes. So we did a test cross. You have the parental types and the non-parental types. You have the total. Be comfortable with this. Okay, let's see here. How are we doing? Okay, if you did not answer D, be comfortable with this. You will get these kind of questions. This is simple recombinant distance. All right, that's just a heads up. I'm not trying to criticize. I know we talked about a lot of different crazy things, but be comfortable with this kind of question. So we just took the recombinants, the non-parental types, and divided by the total. So 47 divided by 100 is a 0.47 recombinant frequency. We didn't correct it with all these mapping functions. So this is the uncorrected distance. Okay. Next. Okay, here's the calculation for D. Are these on the same chromosome? These two genes. So it looks like there's a lot of recombinants. Are there enough recombinants to say they're on different chromosomes? Or are there too few recombinants? It looks like they're on the same, they're linked. How would you answer this? How would you test? Did I hear it? No? Okay. Carrie-Ann Matsuo. Are you? Okay. What would you do to test this? Pass. Okay. All right. What common test are we talking about? If complementation establishes the same gene or not. Yeah, we're just talking about linkage. So it's not. You could try. No. A statistical test. Chi-square, okay? You can use the chi-square test to establish if something is linked or not, is skewed from linkage. That's how linkage was first discovered. If you go back and look at Punnet's results with DF2, there were missing categories that were non-parental. Okay? So our expectants, if they're unlinked,